was invited to give a talk 1.5 years ago, 2 years ago, on our bizarre viruses. And I was sure that we get the most bizarre viruses when she started to speak, and these things are even more unbelievable than what we do. And then immediately when I came back, I decided that I will work on this because this is really mad. And I tried to invite her since then, and I'm happy that finally she does it, because this is a great thing, so different, and we need to integrate what it is, because it's impossible that just plants are doing this. Probably plenty of organisms get these bizarre things, and we need to work really on it. So we're trying to work on it and to see if there is something else in plants that gets this beautiful work. So, welcome.
Thank you. Yes, it was very interesting when we went to a meeting in Costa Rica. It was an EMBO workshop for students. It was training students that came from Central and South America, and it was on viruses, plant viruses. And so Didier and I were in the same session. And I don't know if there was somebody else in that session with us, but I just remember that he was working on the largest viruses, and I was working on the smallest pathogenic agents. So it was very interesting, the contrast between the two talks. So thank you for inviting me. I'm sorry I didn't come last year. It may have helped with the project, or I could have come earlier. But I'm here, and hopefully I can help with your research. Okay, this is tablespoon tuber viroid. That's the theoretical structure. It looks like a double-stranded RNA with loops, because it has a lot of self-complementary base pairing. So that's what we think of when we think of a viroid. This one is 359 nucleotides, very small RNA. But it's not completely double-stranded. It has lots of loops. This is where we're located. This is Beltsville, Maryland. It's right outside Washington, D.C. This is the campus. It's the largest campus that the USDA has for research. We have 4 institutes: plant sciences, which I'm in that institute. We have animal and natural resources, so we have a group of people who do veterinary diseases. We have natural resources, so they work on water quality and quality of the Chesapeake Bay. We're very close to the ocean and the estuary of the bay. We have a human nutrition center that does a lot of feeding studies for not disease, but for health. So they test a lot of things like cinnamon and different things. So they have studies where they pay people to eat, and then they take blood and do other things. And then we have the U.S. National Arboretum. An arboretum is where you have trees and shrubs, and so they do a lot of breeding of trees and shrubs and release those to the benefit of people. So the outline of my presentation, I'm going to talk about viral discovery, classification, biology, structure, something about evolution, because that's very important for agriculture. Some differences between viroids and hepatitis delta virus that was worked on many years ago, and a new method that actually you're aware of here of detecting viroid-like RNAs using deep sequencing. So if you had no idea, there was no homology in the database, you were looking for something totally new that had a circle, like a viroid, you might be able to use this technique. Okay, so viroids cause a number of agriculturally important diseases worldwide. Potato spindle tuber viroid occurs, now it doesn't occur very many places because it's quarantine organism, so you have to test for it. Avocado sunflower virus, chrysanthemum stem, so that's the one we have here right now in the lab. Apple scarskin occurs worldwide. It's also one that's has to be tested, so if we export apples to Japan, for example, they don't want anything, any diseases coming in, so the apples have to be certified to be free. Coconut Kidang viroid shows it, actually, it will kill the tree. There are early stages, and this one they're going to start testing again because it will kill the tree. So we're going to start testing germplasm palm from around the world to see where it's located, and if any of that is kind of ornamental palms that are coming in. Citrus hexacortis viroid is also worldwide and on a quarantine list. So how do viroids spread? So you saw it was a little RNA molecule, and I'll talk more about the biology. There's vegetative propagation, so cutting. So a lot of plants are propagated by taking a cutting. Chrysanthemums, you take a plant, you take the little piece off, and you grow more of it. Grapes, things like that, they're propagated, that's how you make more of it. And so the plant is infected, and of course you make lots of other plants. Mechanical transmission, so the early days with potato spindle tuber virus, the potato field was on the tractor wheel and on the plows. So if the plants were infected over here, these over here would be infected if there was any contact. Because it would cut the potato, you'd have some wounded, and that sap would go in, and you'd have transmission by hands, handling, going in the greenhouse, cutting something, touching it, cutting, and moving over here, you could transmit. Seed, some are seed and pollen transmitted, so you can get seed that you think is virus-free or viroid-free, it might be infected. Insect transmission, not normally. In some cases, there's one case where it was transmitted by an aphid by encapsulation in a virus. So the plant was infected with potato leaf roll virus, and the viroid became encapsulated in the virus, and the insect transmitted it. But it's not a general thing. Most of it is just mechanical transmission. So it's different from a lot of viruses and that does that. How do we control it? Disinfecting tools. If you're in a commercial greenhouse, for example, you would disinfect your tools when you're going through plants. Cleaning the pots, the benches, so treating them with some chemical. Removing diseased plants promptly because they can serve for infection. And actually the best way to control the diseases is making sure you don't have any. So the exclusion of viruses from planting materials by certification quarantine. So we have a lot of materials that come to the United States that growers want to use for making they want germplasm like in France. They like that grape, they want to bring it to the US and use it in their programs. It has to go first to a quarantine facility, thrown out, tested for everything, and making sure it's before it's released to the growers. That's the best way to control it. Okay, so in Europe you have EPPO, which is the European Protection Agency. We have APHIS. It's our Animal Plant Health Inspection Service. And they determine they have a list of quarantine pathogens for viruses, viroids. I don't know if they do it for animal diseases. So they regulate the importation of plants that may contain certain viroids. Potato spindle tuber virus in Europe is a quarantine pathogen. Because if you import seed potatoes from somewhere you want to plant in the field to get potatoes, you have to make sure it's free of viroid. It has to be tested before it's shipped. That's the only way to keep it. So in Europe, you don't really have it. We don't have it in the United States. You don't have it in Canada. Russia has plenty of PSCD in the field because they don't do the testing. They don't have certifications. So the countries really have to develop certifications. Okay, so it was this was 1923. There was a disease in I think in the United States where they found these spindly tubers like this. They were long, and the plants were dwarfed and kind of dark green. And they thought it was, for many years they didn't know what it was. They thought it was a virus. Okay, so in 1923 they saw it. In 1971, Ted Diener and some other groups found out that it was actually a small RNA molecule. It wasn't a virus, it was a naked RNA molecule. In 1978, the sequence was determined the old way, not the way we do it now. In 1979, polyacrylamide gel electrophoresis was used to identify viroids because there's no protein, and I'll talk about the protein associated with this. You can only look for the nucleic acid. So there was in Canada, a gel electrophoresis system, which I'll show you, was developed. Hybridization in 1981 made it very easy and large-scale monitoring and diagnosis of plants. So hybridization technique was developed in our lab. In 1982, they determined or proposed a mechanism for how they replicate. In 1983, constructed infectious viruses, so then we could do reverse genetics and try to determine structure-function relationships. So that moves things along. And then 1994, evidence that RNA silencing may play a key role. So how do Lieutenant Deaner, he was in Belleville, or he retired, he's still around. And they worked to try to purify this agent, a virus, and they found that it was transmitted to tomato. So in potato, they had certain levels, not high enough, so when they went to tomato, they increased the titer greatly. They could actually purify the virus. We talked about doing this. You go on to tomato, so you have a healthy plant. Rutgers is the cultivar. Here's the healthy, and there's the virus-infected plant. So the symptoms are, tomatoes is sort of like our mouse, like you have maybe in the we can put a number of viroids on it and get disease, where in the host plant that they're coming from, they were showing disease. So they did electron microscopy and found, this was 1977. So there's the circle. So viroids have two RNA components. One is a circular, and then one you'll find is a linear, like I showed on the, but it's not really linear, it's like a broad structure because of the pairing. It's a closed circle. It doesn't have a protein component. It's not encapsulated. It's just a naked RNA molecule. It doesn't require a helper virus for replication. So they're smallest agents of infectious disease. I think I'm right about that. No helper. They're highly structured, however, and self-complementary, and that's why you get the base pairing, and that's important for the two groups of viruses that we talk about as a function. And they're composed of quasi-species populations of molecular variants in the plant. So you can find lots of sequences. You may have a dominant one, and if you go to maybe a higher temperature or to another condition, one of those species may predominate, but you have actually a lot of variants within them. And especially true in plants and trees, things that are around for a long time, grapevine, you'll have lots of variants. So what is the difference between a virus and a viroid? Well, like I said, they're not encapsulated. There aren't any virus-like particles, like you might see by microscopy if you're looking at infected plants with a virus. You're not going to see any by usually. There's only a single species of low molecular weight that's required for infection. They don't encode proteins and they replicate autonomously. Well, they use the host machinery for replication. So this is the 2D gel I talked about, the two-dimensional gel that we used initially to look at viroids. If you had no idea, you had no sequence, you didn't know if there was a viroid in your plant. So what you would do, you'd have extract enriched fractions using lithium chloride, using different chemicals. You can enrich for smaller RNAs. Run these on a polyacrylamide gel containing urea at a low temperature. And so you have electrophoresis here, you have healthy RNAs here, and you'll have down here we get the circular and linear component of the viroid infection. It's really in the second dimension where you reverse the electrodes. So now you're going to go in this direction, and you're going to heat it. So we had to make special gel, special plates that have water running through hot water. It's attached right adjacent to your plate where your gel is. And you run it in that dimension, and the heat opens up the circle. So now the viroid opens up and it's a circular RNA, and it runs slower, just like in plasmids, where you have a so it runs slower, and so it separates out. So you can see, this is healthy. Here's a healthy. So these are healthy nucleic acids. Here you can see that circular RNA. So you know you have something viroid-like. Now different ones with different molecular weights are going to be different places on that gel, but they will certainly separate away from all of that. And then we, extract that, cut it out, and then we do random priming, cDNA libraries, just to make, to try to get the sequence when we had no idea. So, okay, since 70, there were like 40 RNA species have been, or viral species have been identified so far. And most of them have been identified because they cause disease. They're agriculturally important on a certain crop plant, or when they're transmitted to a crop plant from another plant that doesn't show symptoms, they cause disease. So So the two hospiviridae, the potato spindle viridae is the type member, that's why it's called hospi. And then there's the Afsunviridae, and that's avocado sunflower viridae. That's why it's called Afsunviridae. The hospiviridae have a central conserved region, and I'll show this later. It has no hammerhead ribozymes, and these replicate the nucleus. This group of viroids has no central conserved region. It has a ribozyme for cleavage of the RNA during replication, and it replicates in the chloroplast. And they replicate by different methods. So this is a hospiviridae. So I showed you before, potatoes and the viroid looks like this. If you do a model of the secondary structure, a computer model of the, we don't actually know. This is just a model. And this is the Afsunviridae. This is peach-late mosaic viridae, which is related to, it's in this group. It has a highly, has a lot of stems and hairpins in the structure. So the structure is really complicated, but all the viroids in that group have this kind of structure. So this shows the list, partial list of, so most of the viroids that we've identified replicate the nucleus. They're in this hospiviridae family, including citrus, columnea, latent. So if it says latent, that means that it's in its host. It is host that we discovered is first. It doesn't cause any disease. If we put this on tomato, it stunts the plant. Or potato, it stunts the plant. Chrysanthemum stunt. You have here. Viricini, this is ornamental. Pepita, citrus, or different citrus, but hop latent is another one that doesn't cause symptoms in hop. There's some coleus, which is an ornamental, it's seed transmitted. Grapevine has several. There's Quinang. Grapevine apple has 3 or 4. Citrus has a lot. Tomato has a few. Apical stunt was discovered in the Ivory Coast of Africa. Tomato plant macho viroid is from Mexico. It's very severe symptoms. And here are the 4, and there may be more, 1 or 2 more now that are proposed to be in this group. That's the Afsunviridae, which is avocado, sunflower. But most of them discovered, maybe that's because that's what we look. There may be more, and I expect there are. These are the replication mechanisms. So it's a rolling circle replication mechanism that uses in the nuclear encoded for the hospiviridae. It's asymmetrical and it uses the DNA-dependent RNA polymerase II for the host. For the Afsunviridae, it uses the nuclear-encoded chloroplast RNA polymerase. So the viroid itself doesn't make the RNA polymerase, it uses one for the host. In the hospiviridae, cleavage and ligation, so it makes greater than unit length multimers during replication. So these are long, you have a unit length circle, which is 359 nucleotides. During replication, you make these longer than unit length molecules, a plus and minus strand, which is really important later. There's a cleavage and ligation that occurs by using host enzymes. In the Afsunviridae, there's a cleavage, but it encodes a ribozyme. So it's a self-cleavage, it's a hammerhead ribozyme, there's ligation, and then it makes another circle from the minus strand. So that's why it's symmetrical. It has 2 circles. The hospiviridae only has 1. But it also has unit 1 greater than unit length RNA, double-stranded. These are completely double-stranded. When you melt, so they're different conformations of the RNA molecule that are important for functions. So this is the double-stranded light monomer. When you heat it, start heating it, the left side opens first, and you have the formation of these secondary hairpins. So hairpin 1, 2, and 3. Secondary hairpin 1 is proposed to be important for movement into the nucleus and cleavage and ligation. It's in that central region. Hairpin 2 may be involved in replication. Then you heat it more, there's hairpin 2, and then you get the circle. And you can see these by electron microscopy. There are other branch structures. This is the high-U branch absent varity that I talked about, and it has regions that are conserved for hammerhead viroids in the plus and minus strand. It has loops. It has this thing called the pseudoknot or kissing loop. They're all important for folding and the function of this RNA for replication or cleavage. They're all important. All these structures are important. So you have to maintain the structure. Over the years, we've mutated almost every base of potato spindle tuber viroid, and we know that we have to maintain certain structure. Because when we make a mutation and it's not infectious, and we make a complementary mutation, we restore the infectivity. So we know that some of these regions are very important. Okay, this is the central conserved region, this middle portion, that can make terpin 1, and that's important for processing and interaction with cellular components. How does it move? So viruses have to move in the plant. So you have the initial infection would occur on a leaf. Let's say you do inoculation, right? So somehow you inoculate on the leaf. And now it's going to move in the plant to make a systemic infection. So the first place it goes is to the roots, with the phloem. That's carries nutrients in the plant. That's the first place you find it. And then it starts moving up to the top of the plant. Okay, then it's got to move from cell. So how does it do that? It has to move from cell to cell. Well, plant cells have cell walls, unlike animal cells. And they have plasmodesmata, certain special connections, and they're different between different cells. So the viroid has to move through different cells in order to make a systemic infection. And it has to go into the phloem and exit the phloem, and all these connections are different. So the viruses, plant viruses encode movement proteins. Most of them encode movement proteins. They're not all the same. They don't all have the same sequences, but the virus needs it to move in the plant to interact with cellular components. The virus don't have it, but they use two host proteins that have been identified that may facilitate movement to help it move around. One is a lectin protein present in the phloem, and there's another one of viroid interacting proteins people have identified. Okay, the viroid replicates in the nucleus. So how does it go into the nucleus? It's not really passive diffusion through the nuclear pores. So we've done studies to use a virus. So we use a virus as a carrier for the viroid. And in this case, we have GFP, fluorescent. We put an intervening sequence in GFP. So if we use this is a plant virus as our transient expression system, we put this sequence into the plant virus. If we inoculate plants with this virus containing GFP, we get fluorescence. If we inoculate it with GFP containing the intervening sequence, we don't get fluorescence because the intervening sequence disrupts the exon, and so we don't get any protein produced, or we don't get functional GFP. If we put, however, the full-length viroid in, and I don't know if I have that here, the full-length viroid, PSTV, inside the intron, we then get fluorescence. So what's happening is the viroid is taking that RNA molecule, the whole RNA molecule, into the nucleus, and the intron is removed. The GFP messenger RNA leaves the nucleus and is translated and makes a functional GFP. So we've used this system to try to identify the region of the viroid that's important for nuclear target. And it ends up being, we haven't taken it all the way down, but we have a region in the upper central conserved region of hairpin 1. May be important for targeting, helping us move. And there's another group working on how do the viroids get into the protein. So they're using a similar system to try to identify the target sequence or structure that allows it to go into. Okay, so the viroid is a collection of structures and sequence proteins. So for different purposes. So there's for cleavage of unit length and nuclear localizations in here. For cell-to-cell and long-distance movement, it's over here and here. For pathogenicity, so symptoms, most of the symptom variants will be big mutations. There are several regions of the virus that are important for pathogenicity. If you go from a symptomless or mild strain of potato spindle tumor viroid to a severe strain, you have maybe 2 to 4 nucleotide differences in this region. That's all you need to go from a mild where you don't see any symptoms to something severe. Okay, so what is it about that region? So this is a pathogenicity region. So there have been a lot of speculation. Is it interacting with some host component you don't know what the host component is? Is it something, when you make those mutations, it's something about the structure? So it's been theorized that there's structure that bends more when you have a severe strain. Or you go from mild, you have this structure, and then you have a different structure. But these are all theoretical structures. We don't know if they actually exist in the plant. But just trying to explain what it is about that, those mutations that cause different symptoms. We don't know yet. We don't know what they are. So this is a symptom of potato spindle tuber viroid in tomato. And when you do micro and macros, you see genes, alpha genes, lots of genes, 2,000 genes. Some go up, some go down. Some of them are stress related because it is a stress on the plant. So it's a general stress response that you would get from any fungus infection, virus infection. So it's not viroid specific. Some are pathogenesis related, some are stress, like I said, some are chloroplast function, protein metabolism, protein kinase signaling, among others. There's a whole list of things that go up and down. And trying to figure out which one is the key has been very difficult. Okay, so we knew, I work on protein phosphorylation, protein kinase pathways, signaling pathways. So we knew in the old literature that the protein phosphorylation was modified in PSTV strains was varying in pathogenicity, resulted in differential activation of the mammalian double-stranded RNA protein kinase called PKR or P68 that Michael Cates works on. Washington and other virologists, animal virologists. And in vitro, it activated this protein kinase. And we knew that there was maybe a homolog in plants using antisera. The group used antisera. It sounds something related. We've never identified that protein. We've never fished out the homolog. Yes, we haven't gotten the ortholog. So, but when we were searching for this one, we did PCR and we did other things. We found another one that was transcriptionally activated in infected plants. It wasn't the one we were looking for, but it was different. There was something. So we studied that one. So these are control, mild, intermediate, severe. So you can see the mild really doesn't have a lot of symptoms, although the titer is very high. So when we look at there's a protein kinase we call it PKV. It's a member of a class of protein kinases that only exist in plants because they have a certain region that the animal protein kinase is going to have. And you have some expression in mild, intermediate, and severe plants. When we make transgenic plants encoding this protein, when we overexpress the protein, we get very dwarf plants. So it's mimicking what we see here when we have transcriptional activation of the plant. When we use antisense RNA and shut that protein way down, plants are very tall, lots of flowers, lots of seeds. And we don't know the function of this in healthy plants. We don't know what role it has in non-healthy. We have a model. This is our model. So PSTV infects the plant. We have overexpression. We have overexpression of PKV. We also have overexpression of pathogenesis of protein, which is a general stress response. PKV is overexpressed. We get more protein production. PKV interacts. PK1 you have is animal cells. This 3-phosphoinositide kinase 1 stimulates autophosphorylation of PKB. This one is important in hormone regulation in the same protein. So we have interaction of these proteins through this domain. This phosphorylates this serine on PKB, which then autophosphorylates, and then we have transphosphorylation of substrate proteins. And we don't know what those proteins are, but they're generally involved in gibberellic acid responses and stuff. So it's a work. Okay. We have evidence that RNA silencing may play a key role in modulating thyroid interactions. That's how they cause disease. We have methylation of homologous DNA. We can detect small RNAs. And this is key to the final thing I'll talk about detection of new viruses. 80% of these small viral RNAs, siRNAs, which you probably also haven't noticed, are derived from a positive strand, although some of them come from a negative strand. You have degradation of partial length recombinant transcripts. The mature, highly structured viral molecule, however, is resistant to RNA silencing. It doesn't enter this silencing complex. So this is the silencing complex. So we produce these completely double-stranded multimeric RNAs during replication. So that's the substrate for degradation by dicers and cleavage that enters this complex, anyway, silencing complex. Here's the mature form. It's not subject to degradation by aldehyde. We have these, they go to the cytoplasm, and then we get directed cleavage, perhaps of some target RNAs, these small RNAs that are produced. It's a mechanism that plants use. Was discovered during virus infections. It's the way that they regulate post-RNA expression and virus RNA expression. So what's the origin of viroids? Well, we don't know that, but there's some speculation that they came from the state intron. They're abnormal regulatory RNAs. There's sequence similarities with SN RNAs. How do they evolve? Well, they're quasi-species infections. They evolve by recombination. At least when you look at sequence among different viroids, you can see that some regions are 100% identical with regions of another viroid. And they look like a mosaic patched together. And it most likely happened when they were infecting the same plant. Upon exposure to selection pressures, which people have done, they evolve extremely rapidly. And they have an extremely high mutation rate. So this was from Santiago, Elena. This is from Spain. They did it was forced evolution because they really forced the viroid to do something very fast. And it was a chrysanthemum chlorotic model, which is one of those highly branched structured viroids. When the calculations they did, they found that viroids have a highest mutation rate among all the other genomes they compared. But again, I would say it was forced. So what are the constraints that operate? Structural constraints, like I said. I mean, you have to maintain the structure of a lot of these hairpins and loops, and that's important. So you can't evolve to something totally new, because you have to maintain some kind of structure. You have to evade silencing mechanisms, so you have to maintain that structure because the structure isn't silent. So you don't want something totally different. There are long distance interactions with some of the viroid sequences. When they fold, we want to maintain those. And they have to replicate. So a key role, viroids maintain in viroid interactions. Okay, so the mature viroids are resistant to degradation. You have to maintain that structure. So epistasis is the interaction between different loci determined in the phenotype, additive and non-additive, synergistic and antagonistic. An approach by the San Juan, and I'll examine all these mutations for stability of secondary structure of 29 viroids that I wanted to see is important. So they looked at the mutation pairs and showed the amount of non-additive epistasis interactions increased with the number of loops. So PSTB was more complex for highly branched than the highly branched Schleicher mosaic virus. So you have a reduced rate of adaptive evolution in different viruses. So the different groups have different rates of evolution. Okay, so what is the role of processing stability of two different polymerases? Because one uses a host-encoded chloroplast polymerase, and the other one uses a host RNA polymerase 2 from the nucleus. So what role does that have in viral evolution? Because we're using two different kinds of polymerases. So although the hammerhead viroids mutate rapidly, they have a reduced rate of adaptive evolution compared to the rod structure because of antagonistic epistasis maintaining their complex structure. What is the evidence for distant plant infections? So there are two studies where people study population of these viruses over the years. Ambrose et al. Found that the progeny of 4 variants of peach-like mosaic virus were the result of rapid evolution. The sequences were heterogeneous, and it could accumulate changes over time. Stable phenotypes gave rise to more uniform than unstable phenotypes. So there's a plan. Sometimes in the field, the plants would be, and we're talking about peaches. So these peaches are in the field for years. And sometimes the phenotype, they look disease, sometimes they don't look disease. So when people go out to the field, they don't know if the plant's infected or not. And because of these instability in the phenotypes and in the sequence. Even though the progeny were heterogeneous, however, and had differences in sequence, structures, and functional elements were preserved. And then that, of course, suggests what I said before, that the structures have a constraint on evolution and diversity. Okay, the maximum number of changes with respect to the parents was higher than that found for PSTB in a similar study in Jamaica. Okay, in 2006, Bedonia et al. In Spain inoculated chrysanthemum stem virus, the way we can work on the chrysanthemum chlorotic model virus in the same host. Those are from the two different families of viruses. The chrysanthemum chlorotic model accumulated at a lower rate, so it didn't replicate to as high levels, but the variability was greater for chrysanthemum stem virus with more diverse secondary structures in chlorotic model. Chrysanthemum stunt, which is a phosphovirus, replicates in the nucleus at a higher rate, and the progenies are more homogeneous. And modeling suggested that diversity was a result of a large fraction of lethal phenotypes not detected by analysis of progeny. And that's, you can have a lot of sequence variants in the population, but over time, a lot of them don't survive, so they're lethal. So can a viroid genome escape evolutionary constraints? So there's a constraint on accumulating mutations because of the structure. You have to maintain the structure. However, you can take pieces of that structure and put them together. So in hospiviruses, there are many examples which I'll show. In absent virus, there's no evidence of recombination between them. Probably because we don't have situations where they're co-infecting the same plant. Hospiviruses can go, most of them can go into tomato. So they could co-infect a plant. So in this case, we have potato spindle tuber viroid, tomato plant viroid, citrus hexacorda. These can all go in tomato. What they found is there are certain regions here, pathogenicity regions, that can, for example, tomato plant viroid may have a variable region from PSTB and perhaps the rod of this left side from the viroid. chrysanthemum or coconut kadang viroid has some variants that occur, they're called slow variants, and they occur in an old infection. So if the plant has been infected for many years, you'll get duplications of the right terminal, this region here, that occurs from polymerase jumping, and it's able to accumulate these mutations. But it's only in that terminal domain. You don't have any changes in the rest of the structure. So that's another way that viral is involved, just by recombination. So it's jumping polymerases, trans-switching. And that happens also in virus infections. Like virus infections where you have co-inoculation. I don't know if it happens in animal viruses, but it can happen in plant viruses. So you can have a mosaic of sequence. So then, of course, you've got a totally new yeast in the plant that if it goes to another host can predominate because you've changed and have some totally new disease or disease agent. Okay, so what happens if you take a plant that's in a viroid population from one plant and go to a different host plant? Well, distinct variants, as I said, it's a quasi-species. There are some that can be host-selected, so they become predominant in that host. And if you go back to the older host, some members of that population can become predominant. The same that were present in the original. So you have fitness peaks and role of tolerance and stuff. So a characteristic of the absent virus, which is that the peach-laden mosaic virus, is unstable symptoms in the field. And you get rapid genetic diversity because you have point change. And so you see variable phenotype in the infection. That's very difficult in agriculture. Because a lot of times people go out to the field. They don't go out and collect hundreds of samples and test each plant and say, is it infected or not? A lot of them are doing by symptoms. So they see the plant and then they say it might be infected. But if the phenotype, if the viroid strain is producing monophenotype, you would never know that plant. So it's a problem for agriculture that you have this evolution. Okay, so what's the problem? So in the top right left, you see Columnea erythra, a lipstick plant. That is ornamental that grows, in the greenhouse. It shows no symptoms, but it contains a viroid called Columnea latent. You put it onto tomato or potato, as I said earlier, and you make severe symptoms. So this is a problem, now an emerging problem in Europe and other places where they're growing tomato plants in the greenhouse. You may have ornamentals. You may have all kinds of things mixed up in the greenhouse. And so they're getting transmission of some of these symptomless viroids into agricultural plants, and it's causing a problem more and more. With expanded seed transmission, could be expanded insect transmission, and the role of agricultural practices in new outbreaks, and that's the greenhouse growers. People who they need to be aware that they have a problem with mechanical transmission moving between greenhouses and plants. And okay, the North Atlantic Plant Protection issued a phytosanitary alert on the first report of Columnea latent viroids in tomato production nurseries in the UK. So Columnea latent jumped onto the tomato in England, and that became a problem for the program. Okay, so now I'm switching totally and just two things related to your project here. Hepatitis delta virus and similarity viroids, and how to discover new viroid-like circular molecules that have no homology to anything in the database. So hepatitis delta virus, and you probably know that better than me here because I've worked on it, was discovered in patients infected with hepatitis B. And I guess in general it makes the infection more damaging to the liver. It's a satellite agent that depends on the hepatitis B virus envelope protein for assembly and daily infecting cells. Hepatitis delta is small single-stranded circular RNA, so small compared to other animal viruses, but large compared to viruses. So 1,700 nucleotides, It has no sequence homology to the helper virus. It can fold with 74% base pairing to form an unpaired rod structure, similar to potato spindle tubercle. So it's a long rod structure. It has a symmetrical rolling circle mechanism, like viroids. It has intermediates of greater than unit length, like a viroid. And they're processed using a hammerhead by design like the F-Sunfera viroids that replicate the multi. So it has a lot of similarities to viroids. So this shows a picture of the hepatitis delta genome. It has a sense and an antisense. The antisense encodes a messenger RNA here, that is capable of encoding two proteins, a large protein and a small protein, that are required for its movement of itself. This is a related kind of viral. It has a plus and minus sense RNA, but doesn't encode any protein. All right, this is showing that there's self-cleavage to make the unit-like monomer. So there's the negative sense genome. There's an editing of the UA stop codon to UGG. So it makes, like I said, it makes a large protein and a smaller integer, which is an RNA binding protein, essential for accumulation. So the question was, where did this RNA come from? Where did it arise? And so there is some speculation that it had something to do with protein. Something to do with protein and the production. But I don't think they know yet where did this thing come from, because there's no sequence homology. And why does it look like viral? Okay, discovering new viral libraries. So for example, you want to look, okay, we looked at animals. There was a disease in chickens. There was an arthritis disease in chickens many, many years ago. Ted Diener used the techniques he knew to apply viruses to look for a virus. But that doesn't mean there aren't any there. So how would you do it if you had no idea what the sequence was? Now, if you were looking, like we do, for diagnostics, we know what we're looking for. We have primers that can detect specific viroids, or we have primers that can detect a specific group of viroids. So if we're looking for a positive viroid, but we don't know the sequence, we can use primers for this conserved region. But let's say you have no idea. It's not a virus, not a fungus, not a bacteria, but you have something. So one way would be to do that two-dimensional gel. That's the way it was done in the old days, where you get the circle. So at least you're getting a circle, and it's a viroid by its circle. It could be like a satellite RNA, a virusoid, they're called, which requires a helper virus, or it could be a viroid that doesn't. So that requires some special equipment, and it requires you to have a titer high enough to see it on the gel. But let's say you don't have that. Okay. So the other way was recently published, but I don't use it myself, was published by a group in California. And it's called Homology-Independent Discovery of Replicated Pathogenic Circular RNA by Deep Sequencing. So of course now we're using the sequencing approach. But you know from sequencing maybe you get millions of reads. So how are you going to figure out somewhere in there is a little viral? Okay. So this technique takes advantage of the fact that in viral infections, at least in plants, you have these small RNAs produced during silencing, the argonaut and there are 21, 22, and 24 nucleotide classes that are produced in an equal from plus and minus strands of the viral generated by dicers. So we have 3 or 4 dicers in plants that do this. I don't know in animals what you have. I read that maybe you have 1 dicer, but I don't know in animals how you do the science. These viral RNAs overlap in sequence and cover the entire length of the RNA by density. Now sometimes there are hotspots, so pathogenicity originates in a lot of them, but actually in the end they cover the whole genome. So that's important. So the group in California developed a unique algorithm, a computational algorithm, to assemble viroid-specific RNAs into full-length viroid sequence in a circle. So this could be used as a potential to discover new viroids with little or no detectable homology. But you still have to show that they have some, maybe, they can replicate. You know, once you get the circle, you don't know how they function. Okay, so what did they do? So they had these small RNA conjugates that they put together based on the viroid, there's the viroid, and these were the known. So PLMV, that's peach-late mosaic viroid, obstinate thyroid, and grapevine Yellowstone viroid were known viroids. And then they had this other thing they found. I think it's called grapevine hammerhead viroid. And that was discovered using deep sequencing and the computer analysis. So they found this new circular RNA. And the steps were to get small RNA pooled. They had non-overlapping small RNAs. They discarded some of those. They had the overlapping ones. There were internal RNAs here. They assembled those. They had these terminal small RNA repeats, or RNAs they discarded. They assembled everything. They discarded. It went through several processes. And in the end, they were able to put together a circle from all of these computer output. Okay, so they had a circle. What is that? So they looked at the predicted secondary structure first, and it looked like the hammerhead, like the absence are, the replicating chromosomes. So it had the blue. I don't know if you can see it. The blue are conserved nucleotides of the plus strand hammerhead that you find, and the red are conserved nucleotides of the minus strand hammerhead. These are the cleavage for the plus strand, that's the cleavage for the minus strand. It had this kissing loop structure that I showed you before. So it looks like one of those chloroplast replicating viruses. They could find it in the plants that they used to isolate. So they could find this using PCR and northern blot hybridization. They found that RNA, of course, in different parts of the plant. But still, does that mean it's functional and infectious RNA? Okay, so they showed actually that the hammerhead structure was functional. So that predicted structure, when they made the transcripts in vitro, they could show that they had hammerhead activity, so they do self-cleave, like you would expect the hammerhead. Okay, so now what they have to do is they have to go back, try to inoculate this thing onto grapevine plants, which is not an unique cell, because you have to do a slash. Grapevine is a woody, so you have to slash the plant, so you got a mechanical transmission and make transcripts into that. So they still have to show one that it can replicate on its own, doesn't need helper virus. Maybe disease isn't important. Just because you find it doesn't make it important. But it is another tool to use, for example, if you're going to look in animals for viruses. It's a good tool. So summary, agriculture, viruses cause significant losses in some crops. Potential now with new, the way we grow things, more challenges to agriculture. We really, in the U.S., we'd really like to find something in corn or soybean or wheat, because then it'd be a real problem. Agriculture, you could get more money. Okay, there are some biotechnological approaches to controlling that have been developed. Antisense RNA, ribonucleases trying to chew it up, but none of these have been adopted in agriculture. For either they don't work well enough, they're not stable, a stable way of controlling. Actually, the best way to control diseases caused by viruses is sensitive detection methods and quarantine regulations. That's the way they control them. Because they're not insect transmitted, they're not sprayed, you just have to make sure you don't have it in the beginning. So that's the best way. The viruses are a collection, like I said, of sequenced proteins. Distinct symptom variants can differ by 2 nucleotides. What does that do to the virus structure? Why can only 2 nucleotides make the difference in having a mild to severe? We're still looking for what the host protein, what are they? We don't know yet what they affect. They evolve rapidly. Silencing is involved in pathogenicity. What's the future? Well, the near future. Identifying silencing targets and pathways and pathogenicities. We're trying to identify those pathways. And we have more data on microarray and where we're focusing on some pathway, and then mostly in hormone crisis. Identifies the signaling pathway, developing control measures, maybe better control measures, increasing the corticosteroid crops. If we were able to use some biotech type of technique, it would be acceptable. It would be acceptable in ornamental plants, but not in food crops. All the food crops that we have in the field, we have millions of acres in the U.S. of corn and soybean and cotton that are transgenic and resistant either to pesticides or to insects. But those are all grown for animals, not for humans. And then we'd like to discover viral species of plants and animals. That's it. Any questions? Yeah.
Yes. Can an infected plant ever recover from these viroids? Can it be cured?
It can be cured by heat treatment. So, for example, some...
That will not kill the plant?
No, it won't kill the plant. You can do heat treatment, you can do meristem culturing, so they do thermotherapy. You can do thermotherapy.
So these viroids seem to be attacking these cultivated plants, preferably, or do you see this in the wild as well?
I think it's just because that's where we see it.
Okay.
I think it's in the wild. We don't know yet where in the wild.
Can they get immune to this?
Can the plants become immune?
Yeah.
Naturally resistant.
We don't know that because we don't know what does that mean, non-host or resistant. Why is a host not a host? So I did some work with a viroid where I used another mechanism to affect the sunflower, which doesn't support the replication of a viroid if you mechanically transmit it. So it went in the group, it stayed, but it only stayed in a certain cambial, in certain cells. It didn't move anywhere. So in that case, things that replicate in certain cells didn't move. So for agriculture, that's not a problem. It can be located. Yeah, so the difference between what's the difference? What is a non-host and a resistant? What makes it a non-host? A Rhamnosus is a non-host.
If you sequence your viroid and obtain different viroid with different pathogenicity,
Yes.
can you use that as a vaccine now?
Cross protection.
Yeah.
That has been used in the past. Yes.
That's what
It works, yes. But then you have
So there's
plants
kind of-
infected with too.
And
But
you
the
think
titer
there's
is there.
competition or
No,
it's
it's competition and the titer is
Competition.
there. But you don't get symptoms, but the thing is still there. So it acts, that's not so good because then it's a reservoir. It's got two things in it. It's not one. But yes, that has been used, cross protection works, like it does with plant viruses.
Can you inoculate this on a plant suspension? If you make a cell suspension, and you just do not,
Such a
just by simple contact.
Very hard, but you can. You can, yes.
So it means it will penetrate by itself. How does it get inside the cell?
Well, in that case, we don't just inoculate, we have to electroporate.
Okay.
You're not gonna, you can't just mix cells in the viroid and get it. You have to get it in there. But you can, but it's very low level. We wanted to do that because we wanted to be able to separate replication from movement, to look at the function of things. And the levels aren't high enough to say, that a mutation makes a difference. You don't have high enough levels of problem.
Did you see any enzymatic activity? How does it replicate?
Do I say what?
Enzymatic activity?
Enzymatic activity with the viroid?
How does it replicate? Does it use?
No, it replicates using the host enzyme.
It is
So
using.
the plant enzymes replicate it, the mod plus strand and minus strand. So it's the host, it's a DNA-dependent RNA polymerase in the plant that recognizes that as a template, at least for positive in the nucleus, nucleoplasm, and the nucleolus. And it accumulates in the nucleolus. The chloroplast uses a different enzyme in the chloroplast. So it doesn't have doesn't do that. Which is why, you know, they seem so simple. They're not that simple. We thought it would be easy when we started making mutations. Like, oh, yes, 359 nucleotides, no problem. And now 20 years later, we're still stuck. We still don't, because we needed the assay. Now we had to be able to separate replication from movement from other things.
Good. Thank you very much.